Okay, so we have two cycles here in week 4, the ovarian cycle and the uterine cycle. The uterine cycle has to do with creating that myometrial layer, menstruation, and things like that, versus the ovarian cycle has to be related to the release of the ovary. Yes, all these dates and all these numbers are very important, but for now, let's just focus on 14. So day 14 is very interesting out of day 28. So 14 is where we have this thing called the LH surge, and this is what triggers ovulation. So let's start off with the ovarian cycle. But before we do that, the cycle length on average is 20 days, but it can vary. So it becomes, I believe, longer with people who are younger, and then it can also become shorter with people who are older or closer to menopause, right? Now, female oogenesis is limited during puberty, and it's a discontinuous process. So what does that mean? Essentially, the cells will divide to a certain point. So they'll go up to meiosis 1, and then they'll stop until puberty recommences, which is quite interesting compared to males, which is often a continuous process. So LH surge, this is the most important thing. This is what is actually resuming ovulation. If you remember, GnRH, the anterior pituitary, releases LH and FSH. So the LH surge is what is stimulating ovulation. Now, why does non-disjunction increase or get worse as our age increases? This is because we have something called cohesions, and essentially what they're doing is they're connecting to the centromeres of the chromosomes, and they're helping pull apart the chromosomes, right? And this is because in our sex cells, essentially we want them to be haploid. We don't want them to be diploid. So if our cohesions weaken, we can no longer attach to the centromere as strongly, or maybe we have incorrect attachment, and as such we have a greater chance of getting non-disjunction, which could lead to things such as trisomy 21, for example. Now, we're going to talk about female oogenesis. So we're going to start from 2N4C to 2N2C, back to 2N4C, and then 2N4C again. Now, we're going to start off with oogonium, primary oocyte, and this primary oocyte is then going to go through this development process of primary follicle to secondary follicle, Graafian follicle, and then eventually an ovulated oocyte. I did want to note though that when we're going from our primary follicle to our secondary follicle, essentially what's happening is that we're reducing the chromosomes, and we're essentially bringing it from 2N4C to 1N2C, right? And all you have to get away from here is that essentially as I'm breaking off the chromosomes and chromatids and making them and getting a haploid cell, I'm releasing these things called buds. And essentially what the bud is it's just material that is going to be degenerated away. And this is quite important in relation to GH, but I won't talk about it now. So essentially you have this budding off, and then these buds are just destroyed later. And the big idea is that as we half the chromosomes, so we get 2N4C after sperm fertilization, we end up losing the DNA. And the proper name for these buds, or these budding off components, is called polar bodies, which are degenerated through likely phagocytosis or something of that nature. So now we're going to talk about the actual follicles, and this will be covered in the GLS about histology, which is quite important because it could come up in an MSAT or even in short answer. So we're going to start off with our primordial follicle. This is essentially going to be a primary oocyte with simple squamous cells. So it's going to look something like this. You're going to see this dot here, and there might be just a single layer of very thin cells there. And then we're going to go off to our primary follicle. This is going to be very tiny too. Our primary follicle is going to be a little bit larger, but not that much larger. And it's also going to have a primary oocyte. However, now the cells have turned from squamous, very thin squamous cells to these cuboidal granulosa cells. And you'll end up noticing that it might seem like it has a thicker ring around it, but there's only one ring. Now, the secondary follicle, I like to think of it as secondary, so therefore two or more rings around our primary oocyte, is that we're now going to have stratified cuboidal granulosa. So we had simple cuboidal granulosa cells, now they're stratified. So stratified just means a lot of them. And now essentially they can be recruited by FSH. And these multiple layers, mainly the outside layer of all these cells, is called the zona pellucida. And essentially these are granulosa cells that are around. Then we're going to get this thing called the late secondary follicle. The only difference is that one, we're going to have fluid form in between the granulosa cells, and we're going to also have these cells called thecal cells form. And what thecal cells are going to do is they're going to promote the production of androgens, and they're also going to produce androgens themselves. And then our granulosa cells are going to be converting the androgens to estrogens. Remember, androgens could be like testosterone, because we're talking about the female reproductive cycle. As per our knowledge till now, testosterone is really not that helpful for our folliculogenesis. So we just want to convert all the androgens to estrogen. Hopefully that is a little easier to remember. But also by doing so, we're also increasing our inhibin. And essentially what ends up happening is that as we're converting more androgens to estrogens, we're increasing inhibin. What is inhibin doing? It's acting negatively on the anterior pituitary. And so what ends up happening is our FSH actually decreases. And what this ends up doing is it chooses a dominant follicle. So you're actually going to have lots of secondary follicles and late secondary follicles kind of competing with each other. And only after that decrease in FSH is one dominant follicle chosen. I believe this is kind of just a safety mechanism where, let's say we have 5 or 6 cells developing, and so now we can pick the best one out of that cell versus having only one cell develop, and then turns out that cell is not that good. So I think that's kind of just like an evolutionary trick to ensure we have the best of the best. Now we're going to talk about our antral cells. I guess the easiest way to explain preantral and antral is just fluid. So if they're big and they have fluid in them, think of them as antral cells. And then, so essentially we're going to have our tertiary cells. So after late secondary, we're going to have tertiary cells. And remember, in the late secondary, we had fluid fill in between the granulosa cells. Now the fluid's going to be filling in the center. And essentially our granulosa cells on the outside are going to attach to the zona pellucida, which was right here. And they're going to form this new protective shell, which is just called the corona radiata. Now, what's going to end up happening is we're going to talk about our Graafian / pre-ovulatory follicles. These are not our tertiary follicles. They actually are, but the main difference is that they get way bigger. So a Graafian follicle is around 2.5 centimeters on average. You can actually see it with your eyes on the slide of the microscope. Now, what ends up happening is they get very big, they get more fluid filled, more development occurs. I would really recommend reading the pre-reading for this week. It really goes into a nice bit of detail and depth that really does help. Now what ends up happening is that our cell was in that, I guess, fluid cluster, and it ends up getting ejected out because we don't want the whole thing to come with us. And this leftover component is called the corpus luteum, and then we just have our cell. So what the corpus luteum is doing is it's releasing LH, and the LH surge is what is causing ovulation. But notice that the LH surge is not from the corpus luteum. This just has to do with this inhibitory effect of estrogen releasing inhibin, decreasing FSH, and eventually this negative feedback mechanism turns into a positive feedback mechanism, which creates this massive LH surge on day 14. So the LH also forms the corpus luteum itself, as in LH is what the corpus luteum needs to be producing all its hormones. And the corpus luteum, the best way I can think of it is it's kind of like the mother of the cell. The cell itself is functionally okay, but it's still requiring the corpus luteum for its different hormones and different effects that aren't really covered. But again, the pre-reading helps with what specific hormones the corpus luteum is providing to the cell. One that I would like to focus on is progesterone. So what progesterone is doing is that it's stopping folliculogenesis. And this is important because now that we've selected our cell, we don't want more cells to start developing, as this could create essentially an ectopic pregnancy or multiple pregnancies, and we want to avoid that. And so essentially what the progesterone is doing is it's acting via negative feedback on the pituitary, And progesterone actually now becomes the dominant hormone because we're no longer having FSH and LH as much, because again, progesterone is acting negatively on the anterior pituitary. So what ends up happening though is, if you remember, is that the corpus luteum itself needs LH. So it ends up just being sustained by another hormone. This is HCG, and this is LH-like. So essentially this is secreted from the syncytial trophoblast after fertilization. So the egg's gonna be fertilized and maybe implant into the myometrium. We're gonna get these syncytial trophoblast cells, and they are what are gonna be releasing the HCG, which is gonna be LH-like for the corpus luteum. What we're doing is we're waiting for the placenta to develop, because the placenta itself can continue producing LH, but it takes time for that placenta to form. Therefore, we use HCG. And this is because the egg, and even fertilized egg until implantation and forming of the placenta, need hormones such as progesterone from the corpus luteum to create a hospitable environment. So progesterone also helps with the myometrium and kind of just creating a very good environment for the cell and implantation and pregnancy and all that. So again, now we've talked about essentially our ovarian cycle. Remember, day 14 is quite important. Follicular days are going to be 1 to 4. The luteal phase is going to be 15 to 28. But just remember, 14 is where we're having the LH surge and our, I guess, ovulation. What we end up noticing is that the secretory phase is days 15 to 28. And this makes sense because before ovulation, from day 6 to 14, you want to have the myometrium ready for the cell, right? Therefore, days 6 to 14 ends up being the proliferative phase. And after day 14, this is where we're getting the secretory phase from 15 to 28. And so let's say after day 28, we didn't have any fertilization or implantation, then we begin our menstruative phase, which is just shedding of the myometrium, So then we're going to have, after the day 28, let's say the menstrual phase, we're going to be shedding of the myometrium. So jumping back to menstruation, it has to do with the wall of the uterus. So we're going to start off with a proliferative phase. This is going to be from day 6 to 14. And the reason I'm starting on day 6 is because it just makes more sense to me in terms of a chronological order. Now, this is before ovulation, so this is before day 14. Therefore, we're rebuilding something called a functional layer. We end up having 2 layers. We have a basal layer and a functional layer. The basal layer always stays there, versus the functional layer is hormone sensitive. It has spiral arteries, and it just helps creating this high vascularized, high blood flow, high oxygen, high nutrient environment for the egg. So after we've built this layer up, and after we have the LH surge on day 14, we end up starting this secretory phase from days 15 to 28. And what ends up happening is secretions happen. So we have lots of mucus, enrichment of the blood supply, and we're essentially preparing for any possible pregnancy. Now, let's say the pregnancy doesn't happen after day 28. We're going to go back to day 1 to day 5, and this is where it's going to be shedding of the old functional endometrial layer. The corpus luteum is going to turn to the corpus albicans, which is kind of this degenerated state. And when progesterone decreases, no longer becoming the dominant hormone, our spiral arteries constrict. This starves the endometrium from O2, and this creates the menstrual flow.